Hello students, welcome back. Today we are going to discuss human reproduction part 7. Today's concept is changes from egg to embryo. In previous part we discussed that egg pronucleus and sperm pronucleus fuse together to form a diploid zygote. It is unicellular and it retains the 46 chromosomes, 23 chromosomes from paternal and 23 chromosomes from maternal. So the unicellular zygote undergoes a number of sequential changes to form a multicellular individual. The stages may take different shapes or forms due to different types of eggs. The basic patterns of sequential changes are zygote undergo cleavage or mitotic division to form morula, and it undergo blastulation to form blastula, and it undergo gastrulation to form gastrula, and the gastrula finally undergo organogenesis and finally develop as a new individual. The cleavage or segmentation immediately after fertilization. The zygote undergoes repeated divisions called cleavage divisions. These cleavage divisions convert a large cytoplasmic mass into number of small cells clustered together, forming a morula. The human egg, being elliptical, shows complete division. Hence, it is called holoblastic cleavage. So let's see that the type of human egg. The type of human egg is elliptical. So there are different types of eggs based on the amount of reserve food material. So that is depending on the reserve food material we will call as yolk. So based on this yolk amount, there are different types of eggs. One, elliptical, that we can see in humans, and second one, it is called as micro lecital, and third one, it is called as mesolecital, and fourth one, it is called as macro or mega or polylecital eggs based on the amount of yolk present in the egg. There are different kinds of eggs, that is elliptical. So which means the negligible amount or no amount of yolk is present. Micro, very less amount of yolk is present. Meso moderate. That means not less at the same time, not more amount. It is a moderate. So more than less and less than more. And macro or mega or polylecital. That means they consist a large amount of yolk present in the egg. So human being, the cleavage is called as holoblastic because due to presence or due to absence of or negligible amount of yolk, the egg or fertilized egg is completely divides into two. Then it is called as the holoblastic cleavage. Now let's see the characteristics of cleavage divisions. All cleavage divisions are mitotic in nature. So all are equational divisions. That means each division and newly formed offsprings can maintain same number of chromosomes. The first cleavage in human zygote occurs in about 24 hours after fertilization. The resulting daughter cells formed during cleavage are called as blastomeres. So that means the first we can say it is a zygote. It is a zygote and it undergo first a cleavage and it has occurred within 24 hours after fertilization and it forms two cells. So mitotic division is equal. So zygote having 46 chromosomes. That means whatever the two cells are formed, each cell get 46 chromosomes. And then again these two cells are undergo cleavage. And it forms another four cells. And it undergo. So like that it will continue till the development of an individual. And now the cells results during this cleavage division. These cells are called as blastomeres. Now, since zona pellucida is intact, not damaged, the size of morula is almost same as that of fertilized ovum. Even though cleavage divisions are takes place, the size does not increase because of presence of intact zona pellucida. Intact means not damaged. As more and more blastomeres are formed, the DNA content increases with respect to the cytoplasm. The first cleavage is vertical and passes through the animal vegetal axis. During cleavage in the zygote into two blastomeres. Second cleavage is again vertical and at right angle to the first one and divides both the blastomeres, resulting in a four-cell stage. Third cleavage is a horizontal division at right angles to the first two divisions, dividing all the four blastomeres horizontally, forming an 8-cell stage. Successive cleavage divisions occur one after another in a highly organized and orderly manner, producing a spherical mass of cells called morula. So the morula is commonly called as little mulberry. So this is the representation of the previous slide. So in previous slide we studied that the cleavage division, first cleavage division, it is initiated on the animal pole, animal pole vegetal axis. So what are this animal pole as well as vegetal pole? So animal pole, that means the egg and yolk, a little amount or maybe negligible amount of yolk or reserve food present in the egg. So that reserve food area, it is called as the vegetative pole, and nucleus area, it is called as the animal pole. Now, so if you are imagine that an axis between that animal pole and vegetal pole, that is appears like this, and the first cleavage division is occurred along with this animal vegetal axis. As a result, the zygote become two cells. The cells are called as the blastomeres. So two cell stage or maybe two blastomeres are formed after first cleavage division. So it is a vertical division. Now these two blastomeres undergo second cleavage division. So first cleavage division is vertical and it forms the two blastomeres, and these two blastomeres again divides vertically right angles to the first cleavage division. As a result, it forms four blastomeres. So this is the second cleavage division. Due to occurring of two cleavage divisions in zygote, it results the four blastomeres, and this stage we will call as the four cell stage or four blastomere stage. And these four blastomeres undergo third mitotic division. So first cleavage is vertical and the second cleavage is also vertical, and the third cleavage is occurred horizontally and it is right angles to the first two cleavage divisions. As a result, it forms total 8 blastomeres, 8 cell stage. Now this cleavage is continue and occurs one after another and form a mass of cells. These mass of cells are called as morula. Already we studied that the morula does not increase its size because it is surrounded by intact, undamaged, intact zona pellucida. As a result, the morula does not increase its size. The next concept is blastulation. As the cells continue to divide further and due to the rearrangement of cells, a fluid-filled cavity appears in the center of morula. The cavity filled with a liquid secreted by the trophoblast cells and the cells of oviduct or fallopian tube is called blastocell, and it is also called as the segmentation cavity. The embryo is now called blastula or blastodermic vesicle or blastocyst. During blastulation, zona pellucida degenerates and allows the blastula in size and volume. Approximately 1 week after fertilization, it is the blastocyst stage that gets embedded into the thickened wall of uterus, a process called implantation, and the pregnancy is said to be established. Let's observe the diagram based on these points. So in fallopian tube, after sperm is penetrated into that, only one sperm is enters into the ovum and the remaining are prevented. So it allows only monospermy, not polyospermy, and it triggers the second meiosis division in secondary oocyte and it become fertilized egg, and the pronucleus of the sperm and ovum fuse together to form zygote, and this zygote undergo first mitotic division to form two cell stage or two blastomeres, and it is a vertical division. And second cleavage division, it is also vertical, it is also a vertical division, and it is right angles to the first cleavage division, results four blastomeres, four cell stage. And the third cleavage is also horizontal division, it is, and it is also a right angles to the first two cleavage divisions, and it results eight cell stage or eight blastomeres. And this cleavage is continue and occurs one after the another to form a mass of cells and surrounded with a zona pellucida is called as morula. Up to this we know morula. Now in this morula, due to the secretion of the fluid secreted by the trophoblast cells. Trophoblast cells, the outermost cells of this morula. So in this mass of cells, the outer cells are called as the trophoblastic cells. The secretions, they secrete the fluid as well as it is also secreted by the oviduct or maybe fallopian tube, and it form as a fluid filled cavity that is called as the blastocell. And during this blastulation, the morula is surrounded with a zona pellucida. This zona pellucida is degenerates. So till formation of morula, it is intact, but during blastulation, this zona pellucida is disintegrates or degenerates. As a result, the morula, after formation of this blastocell, it is called as blastocyst. Blastocyst, and this blastocyst is implanted, embedded on the wall of uterus, that we will call as implantation. So after implantation, the pregnancy is established. The pregnancy and embryonic development. The human zygote undergoes cleavage divisions as it moves down the oviduct towards the uterus. In about 4 to 5 days, it takes the shape of the blastocyst. So we know that the blastocyst is formed through blastulation, and the zygote, it first zygote, it undergo first cleavage to form two cell stage, and the second cleavage to form four cell stage, and the third division, cleavage division, it gives 8 cell stage, and it is continue occur one after another, and finally it become morula, a mass of cells surrounded by zona pellucida. Due to presence of this zona pellucida, the morula does not increase its size even though mass of cells are formed. Now, so this entire process, formation of this, from this morula, the blastocyst is forms. So this entire process, it requires 4 to 5 days. During next 2 days, zona pellucida is disappears. Next 2 days, that is 6th to 7th day, the zona pellucida which surrounds about the morula is disappears, and 7th and 8th day after fertilization, the blastocyst is get embedded in the lining of the uterus, is called as the implantation. As the embryo is implanted, already we said the pregnancy is established. Implantation: the attachment of the blastocyst to thickened inner wall of uterus called endometrium 7 to 8 days after fertilization is called implantation. As the embryo does not have yolk or reserve food, the embryo depends on the nutrition from the mother. The blastocyst has an outer layer of cells called tropoblast and an aggregate of cells or mass of cells at one end called as inner cell mass. Let's observe the diagram. So the fertilization is takes place at ampullary isthmic junction. As a result, zygote is formed. This zygote undergo cleavage divisions and finally form morula. Morula undergo blastulation to form blastocyst. And when these divisions are occur, they are moving towards the uterus in the oviduct or fallopian tube. Zygote is formed at this ampullary isthmus junction, and as it undergo cleavage divisions, it to move simultaneously towards the uterus. And finally, after formation of this blastocyst from morula, on 7th to 8th day after fertilization, this blastocyst or embryo is implanted, embedded in the inner wall of the uterus. The inner wall is called as endometrium. And as we know that the reserve food material is negligible or absent, due to lack of that, the embryo depends on the mother to draw nutrients. So once the embryo is implanted on this uterus, then it is considered as pregnancy is established. So the blastulation, morula to blastula, takes 4 to 5 days, and this blastula implantation, zona pellucida, is disappears in next 2 days, and then next 7th and 8th day the blastocyst is implanted on the uterus. Tropoblast. It is also called as a tropoectoderm. It forms an outer layer of cells or blastomeres in a blastocyst. They make contact with the uterine endometrium and help the embryo to get implanted in the uterus. The cells of trophoblast form protective tropic membranes that later develop into the fetal portion of the placenta, through which embryo gets nourishment from the uterus. Let's see the diagram. So the trophoblast is also called as tropoectoderm. So it is the outer layer of, it is a blastocyst. The outer layer of cells, it is called as a trophoblast or maybe tropoectoderm. So now this trophoblast during implantation contact with the endometrium in the uterus, and in later on stages it become the part of placenta, and which helpful to draw the nutrient from the uterus. So now here see the cavity, it is a blastocell and a trophoblast and innermost cells, the cells which are present to the one end, and another end it is a cavity called as the blastocell. The next part of blastocyst, that is inner cell mass. It is a cluster or aggregate of cells at one pole of the blastocyst or one end of the blastocyst. These cells gives rise to the embryo and differentiate into three primary germ layers, which later gives rise to various tissues of the embryo. After implantation, finger-like projections called chorionic villi appear in the trophoblast and are surrounded the uterine tissue and internal blood. The chorionic villi and the uterine tissue become integrated and form a special structure called placenta. So let's see the diagram. Now, already we discussed the outer layer of cells, it is called as a trophoblast or tropoectoderm, and this later in the development the trophoblast to become the part of placenta, and it helpful to draw the nutrients from the uterus. Now in this blastocyst, one end it consists a mass or maybe aggregate cells called as the, this is called as the inner cell mass. And this inner cell mass in later developments, it is differentiated into primary germ layers are also called as embryonic layers. It develops into primary germ layers or embryonic layers. So these embryonic layers are already mentioned three. So that embryonic layers three are, one is ectoderm layer, and second one is a mesoderm layer, and third one is endoderm layer. From these three embryonic layers, various tissues of the embryo are developed, and the tissues are organized to form as organs, and organs are organized to form organ systems, and organ systems are organized to form as a new individual. So all the differentiates from these three primary germ layers or maybe embryonic layers. So after implantation, the finger-like projections called as chorionic villi appear in the trophoblast. That means the trophoblast layer at this region, a finger-like structures are developed. These finger-like structures are called as the chorionic villi, and these are surrounded by the uterus tissue and internal blood. And this chorionic villi and uterine tissue become integrated, means joined together to form a special structure called as placenta. So now here chorionic villi is a part of fetal or maybe embryo, and uterine tissue is a part of maternal, that means mother. So it form a special structure called placenta. That means the placenta is formed by the integration of uterine tissue, maternal, and chorionic villi, fetal. So both tissues are present in the placenta. The next concept is placenta. After implantation, a special tissue develops between the embryo and the uterine wall is called placenta. This placenta is disc-shaped, temporary connection between the mother and the embryo. Placenta consists of fetal as well as maternal tissue. Maternal means mother. After implantation, the chorionic villi of blastocyst grow into the endometrium. The endometrial tissue undergoes the changes, and the changed endometrial tissue is called as decidua basalis. So now the placenta consists of a fetal part called as chorionic villi and maternal part, that is endometrial tissue, it undergo changes. The changed endometrial tissue we will call as decidua basalis. So maternal part, the decidua basalis, these two are the parts present in placenta. So placenta is formed by integration of the fetal part, the chorionic villi, and maternal part, the decidua basalis. The chorionic villi move deeper into endometrium. They broken down the uterine mucosa, including uterine epithelium, connective tissue, and the endothelium lining, thus coming in close contact with the maternal blood. The blood vessels of chorionic villi are bathed in the mother's blood. This type of placenta is called hemochorial placenta. So let's see the diagram. So the chorionic villi move deeper into the endometrium, that is inner wall of uterus. When it is embedded, the chorionic villi move deeper into the endometrium. When it moves to deeper, it broke down the uterine mucosa, including its epithelium, that means the tissue covers, and connective tissue, and the endothelial lining. As a result, the chorionic villi is come in contact with the blood of the mother, or contact with the maternal blood. So the blood vessels of this chorionic villi are bathed into the mother blood. As a result, the exchange of these substances will take place through diffusion. So here the fetal epithelium is not broken down. So when chorionic villi moves deep into the endometrium, it breaks down the epithelium of the endometrium. As a result, and endothelial lining and connective tissue. As a result, this chorionic villi, along with its blood vessels, are bathed into the maternal blood to draw the nourishment to the embryo. So here the placenta is formed by the integration of that blood of the maternal as well as chorionic villi of fetal. Hence the placenta is called as hemochorial placenta. The placenta is a temporary organ that allows the blood system of the fetus and the mother to come into close contact. The exchange takes place through the membrane, fetal epithelial membrane. Fetus exchange respiratory gases, carbon dioxide and oxygen, nutrients, and excretory wastes with the maternal blood through placenta. Carbon dioxide and waste move from fetus to maternal blood. Oxygen and nutrients from maternal blood to fetal blood by diffusion process. Placenta acts as an ultrafilter, soluble in organic substances, hormones, antibodies against diphtheria, smallpox, measles diseases, etc. Also pass from mother to fetus. Placenta also acts as an endocrine gland secreting a number of hormones. The hormones secreted by placenta. The hormones secreted by placenta are human chorionic gonadotropin (HCG), human placental lactogen (HPL), estrogen, progesterone, relaxin. HCG is secreted by chorionic membrane of the embryo. By maintaining corpus luteum, HCG keeps the levels of estrogen and progesterone high and thereby prevent menstruation and further ovulation till completion of that pregnancy period. Because HCG comes from the embryonic chorion and not the mother, it is the hormone that is tested for in all pregnancy tests. At end of the pregnancy, placenta secretes relaxin that facilitates childbirth by softening the connective tissue of the pelvis. So here we are mentioning that at the end of pregnancy, placenta secretes the relaxin, whereas in NCERT textbook, ovary secretes the relaxin is mentioned. So as per PU board examination, we need to mention ovary secretes the relaxin at the end of pregnancy. So this facilitates childbirth by softening the connective tissue of the pelvis. Mouth of uterus in preparation for delivery. HCG, HPL and relaxin are produced in women only during pregnancy. In addition to these hormones, HCG, HPL and relaxin, in addition to these hormones, levels of hormones like estrogen, progesterone, cortisol, prolactin, thyroxine, etc. increase in the maternal blood. Why these hormones are increased in maternal blood? The increased hormonal production is essential for fetal growth as well as for the metabolic changes in the mother and maintains the pregnancy. Concept is umbilical cord. During the development of embryo, along with placenta, develops another structure called umbilical cord. Umbilical cord is a long and thick cord that runs between the fetus and the placenta. Umbilical cord is attached to the belly of fetus on one side and to the placenta on the other side. Umbilical cord contains blood vessels taking fetal blood to and from the placenta. So that means the umbilical cord consists 100% of fetal blood. That is one most important point. And the placenta consists both maternal as well as fetal. Now, one more thing is that umbilical cord is the connection between the embryo and placenta, not to the uterus. Let's observe this diagram, and here it is a embryo, and this region we will call as the belly region of embryo or fetus. So the umbilical cord connects this belly, one end it connects to the belly of embryo and another end connects to the placenta. So that hard cord present between the placenta and belly of this region. Only the umbilical cord is present. And this umbilical cord helpful to transport the blood between the fetus and placenta. So that's why umbilical cord consists 100% of fetal blood only. Umbilical cord consists 100% of fetal blood. Now the placenta is connects with the uterine wall, endometrium. So when chorionic villi deeper into the endometrium, the epithelium, connective tissue, endothelial lining is damaged and the chorionic villi are bathed into the blood of the mother or maternal blood, and exchange will take place through the diffusion. So from maternal blood, the nutrients and oxygen enters into the placenta. From placenta, the fetus, it is transported to the umbilical cord. Thank you. We will discuss further concepts in the next part.